Hello, Professor. I'm Mubashir Raza. Uh, the year of my registration is Spring 2022. I'm a freshman master student in molecular biology and human genetics, and my advisor is Professor Ingrid Vaidyu. Here is the checklist of my progress report. Uh, my current progress. I have registered the 6 courses, which contains the total credit hours of 6. My timeline: I reached in Sachi University on 25th April 2022. In April and May, I have learned different techniques in my lab. These are cell culture, pipetting, sample preparation, genotyping of mice, restraints test, and animal behavior, open field test, mice handling. In June, I am learning about Western blot. I have learned about the cell culturing of 8022 cells, and here is the confluency of 8022 cells after 24 hours, 48 hours, and at 4 days. I have learnt about the genotyping of the mice. So I have performed the ATF3 mice genotyping, and here are the results for wild type and heterozygous mice. In this experiment, I learned how to give the restraint stress to the mice, especially ATF3 mice. During mice handling, I learned how to handle the mice during behavior experiments and during drugs injection to mice. In Western blot experiment, I learned about the protein analysis and how the Western blot is performed in sleep deprivation mice. Future direction. I have a project. The title of the project is The Effect of Litsi cububa on Pathology of Alzheimer's Disease in Vitro and Vivo Studies. So, what is Alzheimer's disease? Alzheimer's disease is a neurodegenerative disease. It has two pathological hallmarks. One is extracellular deposition of amyloid beta, and second is intracellular deposition of tau neurofibrillary tangles, and it causes the shrinkage of the hippocampus, shrinkage of the cerebral cortex, and enlargement of the ventricles in Alzheimer's disease patient. For my study, I have selected the Litsi cububa, which is a medicinal plant, and its oil, essential oil extract is having the effect of antioxidant and anti-inflammatory. So for these two properties of the LC, I select this as my drug. Hypothesis of my study is LC can improve the pathology of AD in vitro and in vivo studies. In vitro studies, I have selected the 8022 cells, and for in vivo studies, I have selected the 3xTg AD mice. Aim of my study. So specific aim of study is one is to investigate the effect of LC on amyloid beta toxicity in 8022 cells. For this study, I have 3 groups of cells. One is control, second is sham, third is LC, and I will use the different concentration of LC, and after that I will apply the MTT assay or ATP assay to check the cell viability in 8022 cells. In this experiment, I will identify the effect of LC and molecular pathways involved in amyloid beta induced 8022 cells. For this, I have 7 groups of cells, and I first check the after the drugs injection. I will apply the MTT assay or ATP assay to check the cell viability, and after that I will perform the Western blot, immunocytochemistry, and ELISA kit for the inflammatory markers and oxidative stress markers. Specific aim 2 is about the study of effect of LC on learning and memory of 3xTg AD mice. In this study, I have 5 groups of the cells. which each group consists of 8 mice having the age of 6 months, and I will apply the behavior test like open field test, T maze, Morris water maze, trace fair conditioning, and then I will compare the results. Specific aim 3 is to determine the mechanism of LC that regulates the learning and memory in 3xTGAT mice. For this study, I have the 5 groups of the mice. Each group consists of 36 mice of the age of 6 months. I will first apply the behavior test of open field test, tree maze, Morris water maze, trace fair conditioning. After that, I will sacrifice the mice and collect the protein from the hippocampus, prefrontal cortex, and amygdala, and separate the brain tissues for long-term potentiations and ELISA. And I will perform the Western blot and immunocytochemistry staining for inflammatory marker, oxidative stress markers, and for microglial activity markers. Along with this project, I will learn about the behavior tests like the Morris water maze, T maze, trace fair conditioning, tail flick test, and pin prick test, elevated plus maze, passive avoidance test. And the techniques I will learn are the immunocytochemistry, Western blot, and qPCR. I will select the course in upcoming semesters, which covers my 8 credit hours to complete the criteria of my graduation. Thanks all. That's the end of my progress report presentation.